We are going to look at the glycolytic pathway. The glycolytic pathway. And if we look at this, we can have something like this. Glycolysis. And you agree with me that lysis means what? Breakdown, right? Now, glyco here stands for glucose. And the lysis here means breakdown. Which simply means that glycolysis is what? The breakdown of what? Of glucose. So glycolysis is the breakdown of glucose. Now, after taking carbohydrate-based food like rice, like yam, any other carbohydrate-based food. Now, the carbohydrate-based food will pass through the normal process of digestion, right? From your mouth to your gullet, to your stomach, to your small intestine, right? And digestion ends where? In the small intestine. Now, in the small intestine, now the nutrients, the monosaccharides like glucose, like fructose, will be absorbed into the bloodstream. And in the bloodstream, now they can be metabolized depending on the body's need. Now, the glucose in your bloodstream can be broken down to release energy. And that energy we said can be broken down to release energy is actually a broad thing. Is it that? Have you ever asked yourself, you're very hungry, after eating carbohydrate-based food, say rice or yam, is it at that time you just have energy? Or as the food is digested, you have energy? Now, once you get your glucose, digestion have ended, right? Once the food have been broken down, digestion have ended. But the energy is not released yet until this glucose is broken down through the process of glycolysis. Now, the purpose of this topic is to look at the pathway involved in the breakdown of glucose. So we're going to look at how glucose is broken down until energy is released. Do you get an overview of what you want to look at now? All right, so the glycolytic pathway is the pathway involved in glucose breakdown. Let's write. The glycolytic pathway is a 10-step. The glycolytic pathway is a 10 steps multi-enzyme. Say 10 steps multi-enzyme catalyzed linear reaction. The glycolytic pathway is a 10 steps multi-enzyme catalyzed linear reaction that involves the breakdown of glucose to give 2 molecules of pyruvate two molecules of ATP and 2 molecules of NADH Full stop. This reaction takes place in the cytosol. This reaction takes place in the cytosol. Now, if you are done with that, just drop your pen. Don't write any other thing again until I ask you to write. Do me a favor. Now, we said the glycolytic pathway. We've seen that we want to break down glucose to give us energy. We want to break down glucose. Now, the pathway involves 10 steps. And the 10 steps are catalyzed by different enzymes. So that's why we said 10 steps multi-enzyme catalyzed reaction. And the reaction, the pathway is linear. It's a straightforward reaction, not a circular reaction. Do you get that? And it involves the breakdown of glucose to release 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. We'll see how this happens. But for now, just know that in glycolysis, how many steps do we have? 10 steps. Multi-enzyme catalyzed, right? Is it linear or circular? Linear. Now, the pathway involves the production of, leads to the production of how many molecules of pyruvate, how many molecules of ATP, and how many molecules of NADH? Do we still remember the full meaning of ATP? What? Adenosine triphosphate. Why NADH is what? Nicotinamide adenine dinucleotide. The H shows is reduced. Nicotinamide adenine dinucleotide reduced. Alright, so basically this is glycolysis, 10 steps, and we said it takes place in what part of the cell? The cytosol. It is important to note that it takes place in the cytosol, because when we get to the next pathway, we'll see how the product of this pathway gets into another compartment of the cell. So now let's take the pathway. Don't write, just look up. What are we breaking down? Glucose, right? So this is glucose, and the first step in the breakdown of glucose, please don't write, because I'll just be using one-sided arrow. After then we'll correct it back. Glucose is first phosphorylated to glucosylceosate. Glucosylceosate. But now observe this. This is glucose. Glucose have how many carbon? 6 carbons, right? Now this is glucose. This is glucose 6-phosphate. How many phosphate did we add to glucose to get glucosylceosate? 1, right? On position what? On carbon 6. So glucosylceosate does not mean we have 6 phosphates. It means we have 1 phosphate group on carbon 6. So if you are drawing the structure, you have something like this. But don't bother yourself with the structure now, because I want us to finish all the pathways. Then the next time we are covering the pathway, we'll cover them with structures. So for now, just know with their names. So when we are done with all the pathways, we'll come back to each of the pathways, but now not with their names, with the structures, and they are very easy to draw. So this is the structure of glucose, and if you count the number of carbon, this is carbon 1, 2, 3, 4, 5, 6. So you see that there are 6 carbons. Now glucose 6-phosphate, we are attaching a phosphate group on carbon 6. So at this point, we'll attach a phosphate group here. This glucose 6-phosphate. This glucose 6-phosphate. Now, but in biochemistry, we don't do magic. Where are we getting the phosphate group from? From ATP, right? So ATP is coming in. And if we, how many phosphate group are we taking from ATP? 1 phosphate. So how many phosphate group will be left? 2, right? So we'll be left with ADP. Is it not? Remember, ATP, adenosine triphosphate, meaning it contains 3 phosphate group, right? We removed 1 and added to glucose. So we'll be left with 2 adenosine diphosphate. Is it not? Good. Now, we've gotten glucosylceosate, but there's a problem here. The process, why we are breaking down glucose is to give us energy, right? I know the energy currency of the cell is what? What's the energy currency of the cell? ATP, right? That's the energy currency of the cell. We are supposed to be producing ATP, but yeah, we are using ATP instead of producing it. That's the problem. We are using ATP instead of producing it. And when we talk about the 2 phases of glycolysis, you'll see why this is called the preparatory phase or the priming phase. Do you get that? Alright, so from glucosylceosate here, we get to the next step. From glucosylceosate, the next thing here is fructose 6-phosphate. Just understand how it's moving. You'll get it in your head before you leave here. Glucosylceosate to fructosylceosate. Now, what's the relationship between glucose and fructose? They are isomers, right? Glucose and fructose are isomers. So this is just isomerized to this. That's all. Forget about the enzymes for now. We'll talk about the enzymes, but let's just get the substrate. So this is isomerized to this. Did we add any phosphate again? We did it, right? Because this is still 6-phosphate, 6-phosphate. Is it not? Now from fructose 6-phosphate to fructose 1,6-bisphosphate. Good. to fructose 1,6-bisphosphate. Now at this point, from fructose 6-phosphate to fructose 1,6-bisphosphate, what do you notice? How many phosphate group do we have here? 2, right? Because 1,6-bisphosphate. Bisphosphate here means 2 phosphate group, right? One is on position what? 1, and the other one is on position what? 6. So we are having 2 phosphate group. But again, we don't do magic. Where are we getting the second phosphate group from? Again, from where? ATP. So ATP is coming in again and leaving as what? ADP. Good. How many ATP have we used so far? We are supposed to be producing it, but we are using ATP at this beginning part of it. Now, from this point, fructose 1,6-bisphosphate, there will be a cleavage. Please don't draw this because I'm not taking note of the reversible and irreversible reactions. Just understanding is moving before we take that. There will be a cleavage to 2 things. Dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now look at this. Fructose 1,6-bisphosphate is how many carbon? 6 carbon, right? Now it is broken down to 2 molecules of. Now this dihydroxyacetone phosphate is how many carbon? 3 carbon, right? This one is how many carbon? 3 carbon also, to give us a complete 6 carbon. So we've not given out anything. But this is broken down to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now this first phase we have here, what can you say about this? Were we using energy or producing energy? We're using energy, right? So we can call this the energy utilization phase. I'll give you the note. Energy utilization phase, or you call it the priming phase, or you call it what? Preparatory phase. You're preparing to generate energy. You've not generated energy yet. Now, there's a problem here. After this breakdown to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, dihydroxyacetone phosphate cannot continue in the pathway again. He's tired. Or she's tired. So she cannot continue the pathway again. Now we don't have the enzymes to convert dihydroxyacetone phosphate in the glycolytic pathway. And hence, dihydroxyacetone phosphate is converted to another molecule of glyceraldehyde 3-phosphate. Understand this very well now. We have one dihydroxyacetone phosphate here. We have one glyceraldehyde 3-phosphate here. But this cannot continue in the pathway again. So it is converted to another molecule of glyceraldehyde 3-phosphate. It's converted into another molecule of glyceraldehyde 3-phosphate. If I will ask, how many molecules of glyceraldehyde 3-phosphate do we have now? 1 or 2? 1, 2. It's 2 now. Remember, we had 1 already, right? This one is converted to another molecule. Do you get that? Please take note of it, because when we start to calculate our energy utilization, this will be very important. Okay, let me remove this now. We said this cannot continue, right? So it's converted to another molecule of G3P, glyceraldehyde 3-phosphate. So we are having 1 and another one here, right? So we can actually continue the pathway in 2 ways, is it not? But instead of following that stress of writing the same thing year and year, we decide to just put the arrow here. But we will note that anything happening here is happening 2 times. Do you get that? So we have 2 of this here. Now that we've gotten glyceraldehyde 3-phosphate, the next thing is glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. Just follow how it's going. I don't expect you to get it in your head yet. After this, we'll take it again and you must get it in your head. So glyceraldehyde 3-phosphate is converted to a molecule of 1,3-bisphosphoglycerate. But notice something here. How many phosphate group do we have here? 1, right? How many do we have here? 2. Where is the phosphate group coming from? This time, the phosphate group is not coming from ATP. First, how many 1,3-bisphosphoglycerate do we have here? How many molecules of 1,3-bisphosphoglycerate do we have here? 2, right? Remember, we have 2 molecules of this now. So anything we are producing will be 2, 2, 2, 2, 2, 2 until we are done. So here we have 2 molecules of 1,3-bisphosphoglycerate, is it not? Now, the phosphate group here is the other phosphate group we are adding now is not from ATP. It is actually an oxidation reduction reaction. So what is happening here is NAD plus is used to oxidize this to form NADH. Now look at this. There are 2 types of phosphorylation. We have 2 types of phosphorylation here. The 2 types of phosphorylation is substrate level phosphorylation and oxidative phosphorylation. If you notice here and here, we were transferring phosphate from a substrate. We're transferring phosphate from ATP, right, to glucose to give us this, is it not? That is called substrate level phosphorylation. I'll give you the note. But here, the phosphate we are adding is not from ATP. In fact, we are not even seeing any molecule we are taking ATP phosphate group from. Rather, the phosphate group is added through oxidation reduction reaction, redox reaction. So this type of phosphorylation, whereby the phosphate group is added through the electron transport chain, is called oxidative phosphorylation. I will explain more on that. But for now, let's finish the pathway. Now, 1,3-bisphosphoglycerate is converted now to a molecule of what? 3-phosphoglycerate. 3-phosphoglycerate. Now notice something here. 1,3-bisphosphoglycerate. Here, 3-phosphoglycerate. How many phosphate group do we have here? 2, right? How many do we have here? It means we've given out 1, right? And we cannot just write phosphate. We gave it out as what? ATP. So the phosphate group added to ADP to form ATP. Here we produce 1 ATP. Is it not? But remember, anything happening here is happening how many times? 2 times, right? So that means how many ATP did we produce here? 2 ATPs, right? Now from 3-phosphoglycerate, the next thing is what? 2-phosphoglycerate. 2-phosphoglycerate. Did we produce any ATP here? We did it, right? Good. Now I'll continue from there. From 2-phosphoglycerate now to what? Phosphoenolpyruvate. How many phosphate group do we have here? How many phosphate group do we have here? In 2-phosphoglycerate, how many phosphate group? 1, right? On position 2. How many phosphate group do we have here? Still 1, right? Phosphoenolpyruvate. We are still hearing the phospho there. It means we did not release any phosphate group. From phosphoenolpyruvate now to what? Pyruvate. Good. Are we still hearing phospho here? No phospho again, right? It means we've given our phosphate as what? ATP. Now, this is the end of the glycolytic pathway. But if you are asked to draw this in exam and you draw it like this, you might not have half mark. So if the total mark is 10, you might not be given 5 if you draw it this way. Because here there is no enzyme, and in biochemistry enzyme is very, very important. So you must put your enzymes and you must take note of the reversible and irreversible reactions. Here we didn't do that. So we are going to do that. But before then, let's try to understand what really happened here. Look at this whole pathway. How many ATPs did we produce in total from this pathway? 4, right? Good. How many did we produce here? 2. How many did we produce here? 2 again. Because from here, anything happening is from here 2, 2. So we produce 4 ATPs in total. But in the definition, I said we produce 2 molecules of ATP. Is that a mistake? It's not a mistake. Remember, in the priming phase, we used how many ATP? We borrowed 2 ATP from the cell. And overall, now we are producing 4 ATPs. You have to pay back what you borrowed. Do you get? So we use 2 from the 4 we produced to pay back the 2 we borrowed. So we'll be left with a net ATP of what? Of 2. Do you get that? Alright, so basically we are going to take this again and we take note of the enzymes and the reversible and irreversible steps. They are very simple. But if you forget anything we've said today, don't forget anything at all. Don't forget that glucose is first converted to what? Glucosylceosate. From glucosylceosate to fructosylceosate. From fructosylceosate, another phosphate group is added to what? Fructose 1,6-bisphosphate. Now, after that long name, fructose 1,6-bisphosphate, there will be a what? Cleavage to what and what? Dihydroxyacetone phosphate and what? Glyceraldehyde 3-phosphate. Now we said she cannot continue again. Dihydroxyacetone phosphate cannot continue in the pathway again. So it's converted to another molecule of what? Glyceraldehyde 3-phosphate. So at this point, we have how many molecules of glyceraldehyde 3-phosphate? 2 molecules. Now let's take just one. Glyceraldehyde 3-phosphate undergoes oxidative phosphorylation to form what? 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is converted to what? 3-phosphoglycerate. 3-phosphoglycerate to 2-phosphoglycerate. 2-phosphoglycerate to and phosphoenol pyruvate to what? To pyruvate. Alright, so I'm going to clean this and we'll take it again, but this time more attentively. Any questions so far? Good. Don't write this. So it's not, biochemistry is not about writing. In the glycolytic pathway, we are starting with glucose, right? Because that's what we are breaking down. Now, there are 3 irreversible steps in the glycolytic pathway. There are 3 irreversible steps. Steps 1, steps 3, and step what? 10. Those are the 3 irreversible steps in glycolysis. And recall that if I write the arrow like this, is it reversible or irreversible? Irreversible, right? Just one side. But if I write it like this or like this, it shows this is reversible, right? This is reversible. You can proceed on both sides. So anywhere we have reversible reactions, we'll use this or this. I prefer this, but they are both correct. Then where we have irreversible reactions, you have what? So we'll use this. So I said we have 3 irreversible reactions in glycolysis. Steps 1, step 3, and step 10. So this step 1, glucose is first converted to what? Glucosylceosate. Glucosylceosate. And here we said it is an irreversible reaction, right? Step 1. So the arrow remains like this. Now, here we said the phosphate group is taken from what? ATP, right? So ATP is coming in and leaving as what? ADP. Good. Now, from glucose to glucosylceosate, the name of the enzyme catalyzing this reaction. Remember, glucose is 6 carbon sugar, right? And 6 carbon sugars are called hexoses, right? Hexose. Now, one cheat you should know is any enzyme that catalyzes the transfer of a particular group, of a specific group, that enzyme must have the name kinase. If there is a transfer of phosphate group, an enzyme catalyzing the transfer of phosphate group from one compound to another compound, that enzyme must have the word kinase. Now look at this. This is hexose sugar, and there is a transfer of phosphate group from here to here. So what do you think? Hexokinase. That's the name of the enzyme. Hexokinase. Or the more specific one, glucokinase. But generally we use the hexokinase. As we go on, you see the difference between the one that glucokinase can catalyze and the one that hexokinase can catalyze. But for now, hexokinase catalyzes or is a general word. Alright, so from glucosylceosate, the next thing is what? Fructosylceosate. Is it reversible or irreversible? Reversible, right? So fructosylceosate. Fructosylceosate. Now look at this. Glucosylceosate to fructosylceosate. We know that the only difference here is glucose and fructose are what? Isomers, right? So what will be the name of the enzyme? Okay, phosphoglucose isomerase or glucosylceosate isomerase. Anyone is correct. Enzyme that isomerizes glucosylceosate to fructosylceosate. So you can say glucosylceosate isomerase. Or you say phosphoglucose isomerase. They are both correct. Now from here, this is step 2, right? Step 3 is irreversible or irreversible? Irreversible, right? From fructosylceosate to what? fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now, in this fructose 1,6-bisphosphate here, fructose 6-phosphate to fructose 1,6-bisphosphate, we said we are adding a phosphate group, right? So, where are we taking the phosphate group from? ATP. So, ATP is coming in, leaving as ADP. Now, if you look at this, are we transferring a phosphate group? Are we transferring phosphate group? Phosphate group is transferred from ATP to this to form fructose 1,6-bisphosphate, is it not? So, and I said when there's a transfer of phosphate group, what will be in the name of the enzyme? Kinase, right? So, what's kinase? Phosphofructokinase, right? Phosphofructokinase. Phosphofructokinase becomes the name of the enzyme. The next step, we said after this long name fructose 1,6-bisphosphate, there will be a what? Cleavage. So this will split into what and what? Dihydroxyacetone phosphate and what? Glyceraldehyde 3-phosphate. Now, the name of the enzyme that is catalyzing this reaction, this breakdown of this reaction is called what? Aldolase. 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 Now, the next thing we said was that dihydroxyacetone phosphate cannot continue in the pathway, right? So, it's converted to another molecule of what? Glyceraldehyde 3-phosphate. Now, the name of the enzyme catalyzing this reaction is called what? Phosphate isomerase. Triphosphate isomerase is the name of the enzyme catalyzing that reaction. Alright, so the next thing here, from glyceraldehyde 3-phosphate, I'll take it off from here. From glyceraldehyde 3-phosphate now to what? One 3-bisphosphoglycerate. And say what type of phosphorylation is taking place here? Is it substrate level or oxidative phosphorylation? Oxidative, right? So NAD plus is converted to what? NADH. What will be the name of this enzyme? Now, anytime there is an oxidation reaction, you use dehydrogenase for the name of the enzyme. Now, there is an oxidation reaction here, glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. We know this is oxidative phosphorylation, is it not? Now, the name of the enzyme, you can call it triososate dehydrogenase or glyceraldehyde 3-phosphate dehydrogenase. So, let me go with that. Glyceraldehyde 3-phosphate dehydrogenase. Good. Now, from 1,3-bisphosphoglycerate, the next thing was what? 3-phosphoglycerate. Sorry, this arrow, was it supposed to be reversible or irreversible? Step 1, 3, and 10. So this is reversible. This was supposed to be a reversible arrow. Now, 1,3-bisphosphoglycerate to 3-phosphoglycerate, we've given out one phosphate, right? So ATP have been formed here. And what's the name of the enzyme catalyzing this? Phosphoglycerate kinase, right? There is a transfer of phosphate group now from this to ADP, right? To form ATP. So phosphoglycerate kinase. Phosphoglycerate kinase. This is the name of the enzyme. Now from 3-phosphoglycerate to what? 2-phosphoglycerate. Now another cheat. Anytime there is a movement of a particular group within the same molecule, that is, you are just changing position, the name of the enzyme will have mutase. Like in this compound here, the phosphate group was on carbon what? 3, right? We moved it from carbon 3 to what? Carbon 2. So the name, we are just moving the phosphate group. So the name of the enzyme will be what? Phosphoglycerate mutase. Now from 2 phosphoglycerates now to what? Phosphoenolpyruvate. The name of the enzyme here is what? Enolase. And from phosphoenolpyruvate, step 10 is irreversible to what? Pyruvate. And here also one ATP have been produced, right? Good. What's the name of this enzyme? There's a transfer of phosphate group. Pyruvate kinase, right? Pyruvate kinase. Good. So this is the glycolytic pathway. This is the glycolytic pathway. Before I allow you to write and we do the energy analysis and we'll leave this topic for good, don't forget that in the glycolytic pathway, glucose is first phosphorylated to what? Glucose phosphate. Glucose phosphate is isomerized to what? Fructose phosphate is phosphorylated to what? 1,6-bisphosphate. Now fructose 1,6-bisphosphate is cleaved to what and what? Dihydroxyacetone phosphate and what? Glyceraldehyde. The more you say it, the more it sticks. So glyceraldehyde phosphate and dihydroxyacetone phosphate. And we say glyceraldehyde phosphate can continue, right? While dihydroxyacetone phosphate cannot continue. So it's converted to another molecule of glyceraldehyde phosphate. So at this point, we have 2 molecules of glyceraldehyde phosphate, is it not? Now, glyceraldehyde phosphate undergoes oxidative phosphorylation to form 1,3-bisphosphoglycerate. ATP is given off, one phosphate group is given off. And from 1,3-bisphosphoglycerate, we have what? 3 phosphoglycerates. From 3 phosphoglycerate to 2 phosphoglycerate. 2 phosphoglycerate to phosphoenolpyruvate. And from phosphoenolpyruvate finally to what? To pyruvate. Alright, write this down. But sorry, before then, I said from here, this phase here we can classify it as phase 1, right? We are utilizing energy, is it not? I gave you the name the other time. Now, this place where we started producing energy, we can call it what? The payoff phase. The payoff phase, or what? Energy generation phase. Or what? Or the yielding phase. Energy yielding phase. Alright, so you can write that down. Any questions so far? No question.